Hi, welcome to this lecture on environmental adaptation of animals. On this last video on metabolism, we will now discuss on life without oxygen. So why some animals can tolerate these kind of conditions, but we must have constant supply for oxygen. So what happens when we are out of batteries? In normal cell, we have maybe 5 millimole per liter of ATP, and it will be reused and reused and reused and reused. So there's ATP turnover. And this ATP turnover can decrease if we don't have oxygen, meaning that, okay, there's anoxia, or if, for example, in human body, if the temperature in our cells will decrease, so there's hypothermia. And for a short time in anoxic conditions, the ATP production can continue. We have now 3 mechanisms. We can upregulate the glycolysis to maintain it, or we can have this oxygen store like myoglobin in the skeletal muscle, or we can have the oxygen store in the hemoglobin also, so in the circulation there is some oxygen available all the time. And then also we have this phosphate store from where we can put the phosphate to ADP to produce ATP. But sooner or later, these mechanisms are not enough to keep the ATP production. So then the ATP production is lower than the ATP consumption. And that's affected, okay, then the ATP concentration will fall. And what happens? Well, we need the ATP, we need it for protein synthesis, we need it for cell growth and division, but one of the things that is more faster is that we need it for ion gradients. We have learned so far that, okay, we have sodium pump, sodium potassium ATPase, that can consume maybe 30% of the energy need in the cells, and it's working all the time. So one of the things that happens when the ATP concentration will fall drastically is that there is a chronic membrane damages and leak. We can't anymore maintain the sodium and potassium gradient. And this is the place when our neurons are stopped working, because we don't have action potential if we don't have sodium gradient, if we don't have potassium gradient. But also what happens that the potassium gradient is in most of our cells, it's maintaining the so-called resting membrane potential. So if the cell is resting, so beside these action potentials, the membrane voltage difference is maybe minus 80 millivolts or minus 50 millivolts, so something negative. And if we lose this potassium gradient, it will be close to zero. And that affects that, okay, and then there's uncontrolled calcium entry inside the cell. And you know that calcium is a very good trigger of several pathways in the cells, and some of them cells, these pathways are actually causing the cell death. And some of them are rupturing membranes, and that causes the cell death. And in the cell death, there are some fine-tuning mechanisms, but let's not get on those. And in the beginning, when we are having lack of oxygen, we have fine-tuning mechanisms into respiration, but we will discuss those a little bit later during this course. But now you understand the whole scheme that, okay, even the animals that are living without any oxygen, they must keep the ATP concentration quite steady, otherwise they will die. So anoxia means that there is no oxygen, but there's also word hypoxia. And they both mean that there's lack of oxygen, but in hypoxia it's partial. It can be 10% less oxygen than normal, it can be 50% less than normal, it can be 10% or 90% less than the normal. But if there is some oxygen in the environment, it's hypoxic. It can be that it's not enough to keep your ATP turnover, ATP concentration high, but it's still there is something. And beside hypoxia and anoxia, there's a third word you may read in some fixtures. It's chemical anoxia. It's not on the oxygen level, so there's oxygen in the environment, but now this mitochondrial respiration is stopped because, for example, you get some cyanide. Cyanide is blocking the electron transport chain. And if without electron transport chain, there's no oxidative phosphorylation neither. And there's no citrate cycle at all. So everything that happens in the mitochondria on this energy production, it's stopped. And so in both cases, the oxidative catabolism is reduced. And in humans, that causes, of course, a quite fast death. But in some cases, they maintain the function even without oxygen. And this is a very nice study. Unfortunately, there are too many dots. And so they took the crucian carbs in the laboratory and kept them without any oxygen for a few days. And measuring practically everything. Let's take these dots one by one. From the lowest one with the white dots, there's the heart rate. So at the time 0, they started this anoxic exposure. And during the first hours, the heart rate will decrease. So it's beating slowly. But then next day, it's beating again at the same rate as before starting this anoxic period. And then at the in the same lower picture, there is this at blue, also red, there is a respiration rate. So in the beginning, it will start breathing more. The breathing rate is increased, after which it's decreased again within 10 hours because there is no oxygen available. So it tries to catch all the oxygen molecules from the environment. And then on blue, there is a ventral aortic pressure, so meaning blood pressure. So blood pressure is kept quite constant during this anoxia. And then the upper part, there are 4 different color spots, but let's check only on the blue ones. It's the stroke volume that is telling you how much blood is pumped from the heart during each contraction. And as you can see, it's more than when it was having normoxics, when there was oxygen available. So this animal is doing everything else except resting and waiting for recovery. It's increasing respiration. It's increasing the circulation. So it's quite active at that time. So what we can do when we don't have oxygen, we have 2 different strategies. First of all, we have aerobic ATP production that is directly dependent on the oxygen level. We can a little bit upregulate it because by having these oxygen stores, etc. But if we are dependent only on the aerobic ATP production, then the metabolic rate will decrease on hypoxic conditions until it will be 0 and we will die. And in most cases, we are oxygen regulators, so we increase the respiration, but also we increase the anaerobic energy production to fill the energy needs. And this is called Pasteur effect. So we try to keep the membrane metabolic rate constant, but unfortunately, glycolysis is not so effective way to produce ATP. So at a certain time, there is a death of these oxygen regulators. So we can't tolerate very low oxygen concentrations at all. But then the other way is that some animals are oxygen conformers. So they are increasing much less this anaerobic ATP production. So they reduce the metabolic rate because there's not so much oxygen available. And they maintain the metabolic rate at lower, quite steady state, and it's called the standard metabolic rate. So just enough ATP production and consumption to pump the ions, to keep the essential functions inside the cells working, to keep the essential organs working in the animal body, etc. And the division between oxygen regulator and conformer is a little bit too simple. So it can happen also in the same animal. So the animals can upregulate the respiration in moderate, so in mild hypoxia, and reduce the oxygen use in severe hypoxia. And over here we have two different fish. The fish that are in blue dots, they are alive. The one that are in red dots, they are dead. And on the right hand side there is more oxygen available. On the left hand side there is less oxygen available, and in the U scale there is how much oxygen is consumed. And as you can see that when it's not so severe hypoxia, the line is from left right. So they maintain the oxygen consumption quite steady. But then when there's severe lack of oxygen, then the oxygen consumption will drop. And these animals can have differences and when they drop it. So in the top one there's a red line daughter in American fish that is living in fast flowing streams. The exposure to mild hypoxia is actually fatal. So even before there's 50% of the normal, they can't tolerate it anymore, so they are reducing the oxygen consumption. Whereas this other animal, it's actually tolerating quite well 20% of the normal oxygen concentration in the aquatic environment before dropping the oxygen consumption. But in both cases, they can't tolerate very low oxygen concentrations at all, or lack of oxygen. But we have animals that are tolerating it. So some animals that are surviving long time in anoxic conditions. And so usually animals like even ectotherm like rainbow trout, it can last maybe 10 minutes without oxygen. But these anoxia tolerant vertebrates, they are completely in different scale. They can last for 10,000 or 100,000 minutes. And in both cases, you can see that there's a negative correlation between the time you survive and the body temperature. So in cold animals, the cold animals tend to live longer in conditions where there is no oxygen available. And why does it work so well? Well, because in the cold body, also the energy consumption is lower. And how they do it, they actually, quite often these anoxia tolerant vertebrates, they downregulate the activity, the metabolic rate in cold temperatures. Like this chrysalis turtle, red ear turtle, it's the heart rate is about 5 times per minute in +5 degrees of Celsius. So it's been reduced already in normoxic conditions. So it doesn't have to reduce it so much during the anoxic period. Or the crucian carp is also, it doesn't even try to keep the metabolic rate in the same level as was during the summer, but it's reducing it for the winter time. It doesn't eat at all, and etc. So it's reducing it even in normoxic conditions, so it doesn't so much modify the metabolic rate during the anoxia. And also what happens that when they don't have the oxygen available, they only have moderate or mild compensation from the anaerobic metabolism. So they don't want to even try to maintain the high ATP consumption. But they will reduce everything that they have in the life. Okay, ain't that a little bit like metabolic depression? And that's something that also happens in the vertebrates, in the mammals. So one thing that happens in hibernating mammals is also that, okay, they will reduce the energy consumption. We will discuss on the hibernation of mammals a little bit more on the next chapter, but they, even for example bat, they reduce it by 90% when they are hibernating, lowering the body temperature, etc. But still, we mammals, we are not very good in energy saving. So even when we are having this hibernation, it's still 5 times more than same size ectotherm is using. So we are using so huge amount of energy. So even when we are reducing it by 90%, it's still quite a lot. But some animals, they are able to reduce the metabolism almost completely. And a classical example of this is embryo of blind shrimp. And it's normally the metabolism is about 6 millijoules per gram per second, and it's reduced to almost to zero. But it's almost to zero. So there's always some ATP needed, for example, to prevent the drying. So when we are hibernating or depressing the metabolism, the problem is that we are not anymore controlling anything. And that can be fatal. And how this Artemia is doing it, it's very simple way. It's producing lactate and getting more acidic the intracellular solution, and that will keys the metabolism. And this can be tested that, okay, during the after 10 hours they were increasing some ammonia to get the pH a little bit higher, and seeing that, okay, there is some heat production. But okay, then it means that surviving without oxygen or getting hibernated or stopping almost completely the metabolism is actually causing problems in dehydration. And one of the best survivors in dehydration is definitely this guy, Waterbird, meaning karhukainen in Finnish. So these are tolerating dehydration, life in space, and or etc. Very well by having two modes. So they have active mode or then these ton modes, to which they form when they are lacking oxygen or they are lacking water, etc. And when these been tested in this proteasome experiment, it's been shown that, okay, there's a lot of LEA proteins, so late embryogenetic abundance proteins, very long and confusing name, actually found first in the plants. And these proteins are stabilizing these membranes and limiting the dehydration that could cause the membrane damages. And later on, it was found that, okay, Similar importance is also in these brine shrimps. So whatever happens, they try to keep the membranes working, or at least prevent the damages in the membranes. But then, what about vertebrates? What about crucian carp? Crucian carp is one extreme in anoxic tolerance. In Finland, for example, it can live 6 months without any oxygen. And usually the fish, when they are having lack of oxygen, they increase the surface area and water flow through the gills. And in crucian carp, the interesting thing is that, okay, in the gills, actually there is no lamelles at all. You can't see them in the cold. But they exist, but they are inside cell mass. So these gills look like some kind of sausages. And if we put the animal in anoxia or hypoxia, the cell mass will die and reveal these lamelles. Now it's 3 days, 1 week of hypoxia, 14 days. So it's revealing these lamelles, and that's increasing the surface area and increasing the oxygen uptake drastically. And actually this means that now, even then it recovers, so it means that, okay, when there's severe lack of oxygen, it's not going to happen by just a clicking, but it will be a slow process, and during which these animals try to catch all the oxygen available there in the pond or in the lake. So they are limiting the time of complete anoxia. Okay, also what happens is that they are limiting the glycolytic pathways. Again, some proteomic experiment that, okay, over here you have all the proteins in crucian carp brains, and over here you have the glycolysis. And these proteomics was describing that they are inhibiting this process, that process, and the produce of lactate. So they are limiting the glycolysis. So there is not so huge pasture effect, because these processes are limited, maybe to 1/2. So it means that, okay, then the glycolysis is slower, but still, when after February there in the 2003 till March, when the oxygen concentration was 0, it was dropped by 90%, these glycogen storages. So still, although they prevent the glycolysis, they are not preventing more than 50%, it's still enough to use glycogen storages quite fast, actually, because the glycogen, it's been cut to glucose, and from each glucose you only get 2 ATP. So the glycolysis is so ineffective way to produce energy, so you need a lot of glycogen, even when you decrease the rate of glycolysis. And one of the things is affected that because actually although the metabolic rate is decreased, this is in goldfish, well, actually meaning the same animal, that crucian carp, and it's in room temperature, so to get it a little bit faster, it reduces the metabolic rate is reduced to 1/4 during the anoxia. But at the same time, actually, the carbohydrate catabolism can be even increased. So as long as there's any oxygen available, these animals can use other sources, proteins or fat. But at the same time, then when they're having not anymore having any oxygen available, then the only energy source is carbohydrate, and that's why the carbohydrate catabolism is a little bit increased. But the good point in these animals is that they store glycogen a lot in their tissues. And if it works, then the problem is that, okay, you have glycolysis and you have problems with the lactate. Well, you don't if you are drunk fish. These crucian carp, they actually produce lactate acid as all the other vertebrates in brains and heart and muscles, etc. But in skeletal muscles happens the following. The lactate is modulated to pyruvate, which is modulated to acetaldehyde, and which is modulated to ethanol. And this ethanol is, because it's not anymore acid, it's very simple to get rid of it. So you have the ethanol in the blood vessels, and it can be diluted in the environment. So the good point is then that, okay, the pH of this fish is not so drastically changed. Thank you.